name is Lady Science. I will be talking about the non-coding DNA mystery and also our Bernard's article. So what is exactly non-coding mystery, DNA mystery? So as referred to as junk amino acid because there were no known functions. But there is some evidence that non-coding DNA plays a role in regulation of gene expression. The existence of non-coding DNA was a problem of the term mystery, given that it had stood for over 50 years of proving that, or at least to find some evidence of activity. And that is why it was proposed that non-coding DNA was a junk DNA. So a little bit of Bernard's evidence that in his conclusion is in the article called Genomic Code, a pervasive encoding molding of chromatin structure, and also the solution of the non-coding DNA mystery. The authors state that the fundamental connection between DNA and chromatin structure, which the genome codes that participate with the encoding and molding chromatin structure, and also focus in the non-coding DNA, or also mentioned as junk or selfish DNA, as he mentioned it in his article. So in which he uses approaches which compare the evidence of chromatography of proteins of a type of calcium phosphate, which separates phosphoproteins, and also evidence from 55 years ago. As well as he mentioned one approach that was called compositional of genomics, based on the short sequence design of the DNA molecules, and also isochores have similar sequences which differentiate amounts of GC levels. Isochore gene spaces in spatial compartments, in which also an evidence of increasing numbers of genes in partial compartments was in which there was correlated with the GC levels of isochore, and the highest level of isochore was called genome core. He also mentioned isochore as a short sequences 3D structure and nucleosome using structures of GC pores, and also isochore of the family L in each family. So his conclusion is that the GC pore and GC rich isochore correspond to LATs and TADs. He uses the new approach using dot plot GC profiling of 100 kb DNA blocks. This technique provides two different structures, long range in GC pore and GC rich isochores. The result of the data is that long-range 3D structures are possible for the molding of flat and primary TADs, and that they were to be visualized in the section of spatial compartments, in which chromatins of flats are absent and TADs are variables. He also gave the evidence that an important property of long structures of isochore is that they define nucleosome as also physical property of chromatin, as he proposed, and also demonstrate that a fundamental role of DNA sequence and composition for chromatin structures represent a part of it, relative to the protein-based structure model, as he mentioned in his article, in which the DNA-based model of chromatin structure has an important implication that relate to the genomic code and also non-coding DNA. So my personal opinion about these advances in our understanding of non-coding mystery and also chromatin structure is that it was important for our understanding about transcription and the long non-coding RNAs, which also originate from the DNA sequences. And also these advances in science is that it leads to understanding of non-coding DNA as well in the chromatin structure, which contain the genetic information and guides the function of the cells. However, there is evidence that a non-coding DNA contains evidence that act as a regulatory element. For instance, when and where genes are turned on or off. So, for example, some regulatory elements can be promoter, enhancer, and silencer. So, which is really interesting on this article, and also that uh, expand my knowledge in DNA sequences and also non-coding DNA. And thank you so much.